discussing energy landscape, gene networks, and then Cox went a lot on discussing larger complex and aggregation, stuff like that. One of the things that has been made, a lot of the understanding, particularly at the molecular level of these systems, has been a new generation of experiments that have appeared that has allowed us to probe things in many different ways. Some of these experiments come under the umbrella of single molecule experiments, actually encompass a large of very, very different experiments. They all call single molecule, from pooling experiments, from fluorescence experiments. They have nothing to do with it. But anything that you try to probe a molecule at individual base, and I think you couldn't have anyone better to tell you a little about. So, the floor is yours.
Okay, thank you very much for your kind introduction. And it's my honor to be here. My first time to South America, and I had a great time yesterday. Too many drinks. It's worth it. So I'm from Illinois. Urbana-Champaign is a small college town about 2 hours away from Chicago. Most people talk about Chicago. And it is an excellent place for doing science and great faculty members and students, especially the physics department. We just published a single molecule laboratory manual by the Crosby Harbor Laboratory Press. And this is what I wrote in the first, the first chapter of the book. So it's biology in singular. So it's pseudo-Latin. So the opposite will be biology in oracle. So many biological reactions are too complex to fully comprehend through the use of conventional ensemble techniques. But at the most fundamental level, all biological reactions occur via the action of single enzymes in their DNA, for example, RNA or DNA molecules. Studying one biological macromolecule at a time, or biology singular, can provide us with an extraordinarily clear and often surprising views of these molecules in action. So that's the motivation that we have for taking this approach. Well, just to break them down into more digestible bits, so why study single molecules? Well, if you study single molecules, you can detect very directly subpopulations and dynamics of individual molecules. This is not truly unique to single molecule measurement, but you can't really think of a more direct way of probing the subpopulations than measuring one molecule at a time. And if the dynamics that you are interested in is not synchronizable, protein folding can be initiated by changing the solution conditions suddenly. So you can synchronize the dynamics, you can measure protein folding beautifully in these ensemble techniques. But there are many of the reactions that cannot be synchronized. And then single molecule measurements are almost unique in providing you the dynamic information. Although I also have to say that if you do careful NMR experiments on certain conditions, you can also measure dynamics if you have the right time scale, even when you cannot synchronize the reaction. Okay, so that alone itself is not unique to single molecule measurement, although it's almost unique. We can also probe the molecular heterogeneity. We can show that nominally identical molecules, if you look at them one at a time, they're not really the same in their behaviors. And after all, maybe it's not so surprising because you're not looking at a simple diatomic molecule, looking at molecules made of hundreds, thousands of atoms. They are quite complex systems. Perhaps we shouldn't expect them to behave all in the same way. But again, the single molecule measurements prove that these heterogeneous exist. And this remains to be seen whether these differences between molecules have biological meanings or not. Finally, it is also very, very interesting to make measurements of single molecules, but not just one number, but multiple properties from the same molecule and make correlations between these molecules. Jose alluded to the measurement of the baseline of the audience in today using this rover, but you can also correlate this with the dietary habit of each person. And then you can learn a lot about the dependence between the two. So the ability to make this a different property, correlation between different properties is, it's almost a unique strength of single molecule measurements. If you think about this, this is the ultimate reductionist approach. Okay? If you study biology, it's hard to break the problem down to even smaller level. So single molecule methods measurements appeal to a lot of physicists and students who are physics oriented. And as a professor teaching in a physics department also, I find it very nice to actually use single molecule experiments as examples when I teach biology to physics students who may know nothing about biology, because these are very easy to understand, very intuitive, very clear and simple experiments that people can do. So it's a great vehicle to through which to learn biology. Two years ago, I was asked by this journal called ACS Chemical Biology to write a review on single molecule techniques in general. So I did what professors usually do, ask a postdoc to write a review paper. But I did spend some time doing this exercise. There's a database called PubMed that indexes all the biomedical publications for the last 30 years. So I went there and then I entered single molecule as a title word for papers, and then counted number of papers with that title word as a function of year, starting from 1989. So you get from 0, and then it is growing exponentially with a doubling time of 2.2 years. So it's really explosive growth. If you look at the number of papers indexed by PubMed over the last 20 years, the same period, it nearly doubled. Okay, so 2.3 years versus 20 years. So if you make a simple physicist-minded extrapolation by year 2030, then every paper indexed by PubMed will be a single molecule paper. But to be more serious, I think it's plausible that perhaps many of the, most of the biological papers by then may contain a figure or panel in a figure that shows the single molecule data, because it's becoming easier to do, and more people are realizing the power of this approach, and it'll become more commercialized and so on, so that it'll just be like one of the routine tools that people use to investigate their biological problems with. So you can categorize molecular measurements into roughly 3 different groups. The first single molecule measurements are done in single ion channels. These are channels embedded in the membrane that conduct current, and those measurements are done using a technical test plan in the 70s that led to the Nobel Prize, and showed that for the first time these ion channels are really switches that turn on and off at the individual molecule level. The second more recent approach is to use mechanical manipulations using optical tweezers, magnetic tweezers, or of course microscope to apply force to measure the response of these molecules, also using the mechanical measurements. So it's force-based measurement, and that will be the subject of today's lecture. And the last area is you can call it optical detection, but mainly done using fluorescence measurements. And this is actually what I do and what my students do in my group. So that will be the subject of tomorrow's lecture. And I don't mean that this is present, this is the future of the field. just today and tomorrow's lecture. But in many of the measurements that people are doing are very, very creative. I mean, it's amazing. It's really up to the imagination of the students who want to, similar lines of attack to problems. So many of the measurements that people do cannot be easily, put into one category. Okay? Just if you go to these meetings, if you go to biophysical study meeting or smaller meetings, it's breathtaking how many different approaches people are taking, incredibly diverse area of research. So what kind of biological problems have been studied using these approaches? There are many, many. So people have studied macromolecular folding, protein folding, RNA folding, DNA folding using these approaches, and also the mechanics of proteins, DNA, and RNA. And many motor proteins have been studied. Most motor proteins are known to use ATP hydrolysis to move on the molecular tracks. And depending on what molecular tracks you're moving on, you can group them into two. One group moves on cytoskeleton tracks, actin filament or microtubules, so that there are cytoskeleton motors, such as thymine, myosin, and kinesin. And there are also motor proteins moving on the nucleic acid tracks. Helicases, DNA polymerases, RNA polymerases, they move on DNA, while the ribosome is a motor protein moving on the RNA track. And membrane proteins have been studied, not very extensively, except for the ion channels. DNA replication, repair and recombination, transcription by RNA polymerase, translation by ribosomes. They are all accessible to these techniques. And more recently, people are starting to do measurements in living cells, trying to study cell signaling. And tomorrow I will touch upon a new exciting area related to super resolution imaging. Most of the single molecule measurements, however, have been done in isolated molecules. You have one molecule in isolation, you measure their properties. You can get very good data, very novel information. But these molecules do not function in isolation in the cell. They work with other proteins, forming a complex. So, so if you want to better emulate the in vivo conditions, then you want to be studying more complex systems with many components. But more traditional way of measuring just one number as a function of time, the distance as a function of time, or current as a function of time, may not be enough if you want to study a very complex systems. You need to gain more information. And so now people are trying to do fluorescence measurement using many different colors at the same time, and also combine different modalities of single molecule techniques, for example, combining fluorescence and force techniques. We also like to maximize the information content of these measurements. So we need to somehow stabilize the instrument and stabilize the signal so that you can look at a single molecule for a long time, and also with higher time resolution and higher spatial resolution. And once you obtain data of a very complex kind, then you don't want to use just eyeballing to analyze the data. You want to have an unbiased way of extracting useful information from a complex single molecule trajectory. So these are the challenges lying ahead. Okay. So my department of physics has about 65 professors, so it's maybe the largest or second largest in the country. And 7 of us are doing biological physics full time, so it's actually usually a large number of people in the physics department, maybe again second largest to UC San Diego. We have 5 experimentalists here, and in fact, I would say about 3.5 of us are doing single molecule measurements. So we of different kinds. So we talk to a lot, each other a lot. And Klaus Schulten and Alexei Xenentiev are the computational biophysicists who they do a single molecule experiment, but not in the lab, but inside the computer. And so they can talk to each other and then compare results. Oh, so I also have a Brazilian connection. So Ivan Brazil is actually is a citizen of Uruguay, but he did his PhD in this place. I heard that this is a place near São Paulo. Yeah, it was an excellent. My first postdoc, and he did really beautiful work. And now he's an assistant professor of Emory University, leading privates like NSF Korea or so on. And now I have another postdoc who was born in Brazil too. Okay, so today I'm going to start by illustrating the power of this approach using this amazing rotary motor as an example, and then move on to discuss 3 different techniques. One is optical trap, and we also have magnetic trap of tweezers, and the atomic force microscope. And I'll give you a few examples each. So the device that I'm showing you here is called F1F0 ATP synthase. How many of you have heard about this? Everyone. Okay. So I'm going to try to explain it just to remind myself what it is, because I have to constantly remind myself. It's very complex. So you have a membrane in the mitochondrial surface, and you want to convert the difference in the protein concentration in and out of the membrane into chemical energy stored in the form of ATP. And this is done by coupling the protein movements from one side to the other with the rotation of this hexamery unit with actually a straightforward symmetry around this central gamma subunit. But the way it actually happens is that you have this device here in gray that holds this guy into the membrane, so that when rotation actually occurs, what rotates is the one in the middle. People have shown and proposed that this is a rotary motor that probably moves 120 degrees per ATP synthesized. And it was shown in a very elegant experiment about 10 years ago that if you simply remove this F1 part of the motor without the membrane part, then you anchor this portion to a glass surface using antibodies or other attachment method, and then look at the rotation of this gamma subunit by attaching a large mechanical load, in this case an actin filament. Then you can actually watch it rotate around the central pivot point under the microscope using a fluorescence imaging. So these are the movies published about 10 years ago. So you can see them rotate. And that was before I started going to the biophysics meeting. So they presented this result in 1997 at the Biophysics Society meeting. But I heard that the room was packed, and when they showed the movie, people started to clap. And then people stood up and then continued to clap. So the first, this is the only example I know where actually somebody got a standing ovation in a scientific talk. And that can happen only once, right? Because if you show the same data, the next meeting, nobody's going to stand up and give you an applause. So it's a really amazing experiment. So one thing that you can notice is that this model, this is rotating much more slowly compared to these two others, and that is because they somehow got a larger actin filament attached to it. So due to the large size, and you get actually larger viscous tract that slows down the rotation. So the speed here is not determined by the fundamental limit of the chemistry, but by the viscous drag. So in fact, they use this method to reduce the conversion efficiency of this motor. When you break down 1 ATP and rotate this by 120 degrees, then how much of a work can the motor do? Okay. It turns out that 1 ATP stores about 20 times the thermal energy at room temperature, 20 kT. But 1 kT is 4 times 10 to the minus 21 joule, and in units relevant to the nanoscale molecules, that the energy is 4 times distance, so scale is about nanometer for many biomolecules. So in this unit, that this corresponds to about 4 piconewton times nanometer. So that means the thermal energy corresponds to, if you want to make a change in properties of molecule by applying force, if the relevant length scale is 1 nanometer, then you need to apply about 4 piconewtons of force to make the change to happen. Okay. So for 20 kT, they're talking about 80 piconewtons times nanometer. That's the energy released by 1 ATP pyrolysis. And you can actually calculate the torque the molecule is able to generate from this measurement. And you know that torque has the same dimension as the energy. Right? And you multiply the angle that you rotate through. Then you can show that indeed, actually, amazingly, this motor protein generates about 80 piconewtons times the nanometer of torque. If you integrate over the torque times angle is about the same scale as the 1 ATP energy. So you're talking about nearly 100% conversion efficiency. And then in the following year, they went to even smaller actin filaments, okay? And so that viscous drag is less of an issue, and they also lower the ATP concentration. And then they were able to see steps. So let's see this one, please. Okay. So what they saw was that the angular positions of this motor were quantized in 3 different directions, about 120 degrees apart, and you can actually see as a function of time the number of revolutions occurring in 120 degree steps. So that was really a really nice demonstration. And then subsequent experiments by them and some others show the following. For example, they ask themselves whether the steps are caused by still the relatively large load of actin filaments. So they attach single fluorescent molecule to this motor, and they showed that yes, you can still get 120 degree steps. And then they went to a sort of intermediate sized probe, a gold particle that is smaller than the actin filament, so it gives you even less viscous drag. And but you can actually measure a gold particle with much higher time resolution than single molecule fluorescence imaging. So with high time resolution, they were able to show that the 120 degree steps are actually made of 2 steps of 90 and 30 degrees. If you go to YouTube and enter this molecule's name, you will see a movie, and it shows 90 degree, 30 degree steps. And later on, they corrected themselves by saying that actually it's not 90, 30, it's more like 80, 40. But that's the details. They also show that by forcefully rotating the unit using a magnetic trap, in the reverse direction, they show that you can actually make ATP, starting from ADP and phosphate. And finally, all the measurements so far had been done on this F1 unit only, without the membrane part that is needed for ATP synthesis in the natural way. There's another group in Germany who actually studied the entire thing, and then they showed that it indeed rotates when you actually have ATP synthesis going on, but in the opposite direction. And also in steps of 120 degrees. So that was one nice illustration of a really famous single molecule experiment. Now, the second topic here is the technique called optical tweezers, optical trap. The idea is very simple. If you have a laser light that is tightly focused down to its diffraction limit, let's say half a micron for near-IR laser light, then due to the gradient force, the particles, small particles that you have in this solution, as long as it has higher index of refraction than the medium, then it will actually suck into the middle of the laser focus. And if you use a stronger laser light, you can stabilize the particle in the middle of the trap better. If you have a weak laser light, then it will be somewhere in the middle, but it will undergo larger volume motion. But it will still be trapped near the middle of laser focus. And this was discovered and reported by him, Arthur Ashkin, 20 years ago in the laboratory, in collaboration with Stephen Chu here, who was my postdoc advisor. And this guy didn't do anything.
Who's more important?
Maybe the next president.
It takes some work. It's going to take some work.
Yeah, still something talking about Alan. Anyway. So you can use it in many different experiments, but this is one famous example here. So if you have the bead microsphere trapped in the middle of the laser trap, and then you have an RNA polymerase making RNA based on DNA template, but it's a monoprotein moving with the DNA. But if you stick that enzyme to a glass surface, then when it pulls on the DNA during the RNA synthesis, then the DNA tether will become shorter and shorter. Okay? And you will be applying force to the microsphere. And so the microsphere will actually deviate from the center, just to balance out the force, restoring force versus the pulling force. And that's the idea. So this way you can, for example, study how the force can regulate the speed of the transcription. We can also study how what kind of force that molecule actually can exert before it gives up. Oh, okay, so I have one here. So there are many things moving in the cell, and as physicists, we love to study moving things. Right? And these motions are done by motor proteins. And one famous example is a protein called Kinesin going on a track called microtubule. And this is kind of artistic rendering of the process based on some data and based on some imagination by Milligan and Vail. And here, it is a motor protein that is a dimer, two identical units are made into a dimer. And using ATP binding and hydrolysis, it is thought to actually walk on the microtubule as a bipedal walker. There is a bit of a confusion in nomenclature in the field because to me they look like feet, right, as a walker, from a walker, but they are actually called heads. Okay? But the mechanism that this animation depicts is called the hand-over-hand mechanism. Okay? It's confusing. So to illustrate this point, I as my others. Sorry. Yeah, let me show you this. So, it's my daughter about 4 years ago. Okay? So she's very good in doing the monkey bar. And so that's one way of doing the monkey bar, hand over hand. Right? And this mechanism is nice because you can actually go off quite a distance without falling off. Okay? And these kind of motors are called processive motors. So she's a highly processive one. Maybe only by the length of the thread in this case. So in fact, just a few years after the optical trap was invented by Ashkin, Stephen Block used this technique to show that Kinesin actually walk in 8 nanometer steps. Okay? And I think there's a really simple way of ending an abstract with a paper. We find that Kinesin moves with 8 nanometer steps. Okay, he's a good banjo player, so I took this movie about 2 years ago. Okay. So these are the movies taken from his website. So in this case, you have a Kinesin molecule moving in the microtubule, but somewhere here you attach a large bead, okay, as a cargo. And then you watch the cargo move because it's actually difficult to just look at the Kinesin itself because it doesn't really give you any optical signal. Okay? So what they did was they attached a really large cargo and then followed this movement. The cargo is about a micrometer size bead. So in this differential interference contrast microscopy, you can see the microtubule as a line. And the bead is moving along the line because of the processive movement of the Kinesin motor carrying the bead along the microtubule. And this is, when you simply observe it without any optical trap. It turns out that there is actually enough of the bead, no trap of the bead, that you can't really see its individual steps. So the trick for them was to use a laser trap to suppress the Brownian motion and then measure the individual steps. So this is what they do. So you have a laser trap in the middle of your microscope, and then you move your sample once until you capture this bead in the laser trap. And then you move the trap toward the microtubule to land your Kinesin carry bead on the microtubule and let it watch. Okay? So what happens here is that as the Kinesin walks in this direction, down this direction, it is moving away from the center of the trap, so there's a buildup of the distorting force that becomes larger and larger. But eventually, Kinesin cannot go past a certain force anymore, and then it'll fall off, and then bead is sucked back into the middle of the trap. Then you can stop the reaction again. Now, although the particles are about a micrometer in size, you can actually measure their center much better than the size of the particle. In fact, if you have a hill, mountain, then you can actually determine the center, the peak position much better than the width of the mountain. Even when you don't have a marker like that. So if you do that, then you can determine the center position with a nanometer precision as a function of time. So this is a more recent study. Again, the Kinesin carrying a bead on a microtubule. And in this study, what they found is that Kinesin sometimes linked can see these 8 nanometer steps, but you have a step that is short in duration, followed by a step that is long in duration, and they alternate. Short, long, and short, long. So it looks like the kinesin is dragging one of his legs, limping along the molecule. And in fact, this is a good evidence that the hand-over-hand mechanism is correct. Because only when you have two monomers alternating in their function, you can have this asymmetry, limping kind of data. And also it's suggestive of intrinsic asymmetry built into the machine, although like in any single molecule measurement, you cannot rule out the possibility that perhaps your measurement itself is inducing the asymmetry into the system. So there's something to be looked at in the future. So in the movie I showed that if the molecule moves too far away from the center of the trap, then it just gives up because it has reached the stall force. Okay? The stall force is basically, when a motor is moving in this direction, there's something pulling it back. Okay? And up to what force can I actually continue to move? Okay? So there's a limit here because, again, 1 APP gives you 80 piconewtons times nanometer. And the step size, you move one 8 nanometer step for APP, so that then the force that you can move against cannot be larger than a certain number, which is in this case 10 piconewton. Okay? So that's the maximum force you can move against, and if you should measure the stall force of kinesin, it's about 7 piconewtons. So that's not actually too far from the theoretical limit. It turns out, I learned, that measuring the stall force is not an invention by a single molecule biophysicists. Actually, it has been done for many different kind of sciences. So this is a cartoon that I stole from Kinesin paper. I forgot his name, but he studies flying insects in Penn State University, and he had a pre-med student who actually did some research. What they did was they study insect flying. So they attach a weight to the body of the insect and see if it can fly. Okay, and if it can fly, then you increase the weight, and then you continue to do so until the flying insect cannot fly anymore. Okay, and you do this for many different kinds of flying insects that span about a couple of magnitudes, order magnitudes in the body mass for the muscle, and you plot the stall force in this case in the log scale, and then you get a nice line with power 1. And then they continue to do so for flying bats, flying birds, and running animals and swimming animals, electric motors, piston engines, all the way up to jets. And they found that everything actually falls in one line. We can also use optical trap to study the mechanical properties of double-stranded DNA. In a very old model called freely jointed chain, DNA can be thought of as a polymer made of n identical units of length L each. Yeah, so this is an example. And in this model, each neighboring segment can actually take an arbitrary angle relative to the other one. Okay. That's why it's called freely jointed chain. And in this, so it's similar to random walk. And if you measure the end-to-end distance, and if you measure it along the direction of applied force, if you actually pull two ends of the polymer and apply force in this direction, and if you measure this distance and you average it over all possible configurations under this model, then you get this equation, which is actually identical mathematically to the alignment, magnetic moment under a constant external magnetic field. So there's a well-known expression. And Carlos Busmante's group in group explained that he was in Oregon about 15 years ago. They managed to measure this basically average Z as a function of force from a single DNA molecule. So the curve is shown here. This is force versus extension, the length of the DNA. And the square symbols are from experimental data. And then they try to fit the data using the Fourier joining chain that I showed in the previous slide. That's shown in dashed line. And it fit the data pretty well in the low force regime, but it deviates strongly in the high force regime. And that is because in that model, you actually ignore the possibility that these molecules can also extend and bend by themselves. So the better model is called worm-like chain model, which has been used very successfully in this area. It has a very simple description mathematically, really. Is if you have a chain of a polymer, let's say you have 2 points, 1 and 2, and at these 2 points you determine the tangent lines and determine the vector T1 and T2, these 2 vectors, unit vectors. And then if you increase the contour length S between the 2 points, then at some point, these 2 angles will not be correlated anymore because of the flexibility of the polymer, and you'll lose the memory of in which direction you're pointing some distance ago. So if you actually determine the dot product between the 2 vectors over a certain separation over the entire polymer configuration, that actually should start from 1, of course, and then decay to 0 over a certain length scale. And that decay, exponential decay, occurs over this length scale called persistence length, LP here. So what it means is that the polymer itself is quite straight up to about persistence length, and beyond that, it becomes flexible enough so that you don't remember the original direction anymore. For double-stranded DNA, that length is about 50 nanometers. Okay? So that's about 150 base pairs of double-stranded DNA. Single-stranded DNA molecules are much more flexible, and it depends on the salt concentration, but it can range between 1 and 3 nanometers. So between about 2 and 5 bases of DNA. You can also use optical trap, not only to study the elastic properties of the DNA, but also to learn something about the folding or unfolding the energy landscape of DNA. For example, in this case, the DNA or RNA hairpin. So let's imagine you have an RNA molecule that has a standard loop structure. It's a hairpin. So you have many base pairs, and you want to understand the reaction coordinate and energy landscape of this molecule. So what you do is you have this RNA structure in the middle, and then you hybridize it to 2 handles of DNA, about 500 base pairs in length. And then 2 ends of these handles are labeled with small molecules such as biotin or reductionist. These are small molecules that have a well-known binding partners that are very stable and specific. Then 2 ends can be attached to 2 microspheres that can be trapped using optical traps. And then you can move the laser beams apart so you can apply the force in this direction so that you can mechanically unzip the hairpin and then see what the molecule does. So what you can actually do is to measure the distance between the two beads as a function of time. But you can also keep the force constant and then see what it does. So this is a result of a measurement done by the Bustamante group. So the data here is obtained at a low force, 13 piconewton. So the hairpin is mostly really in the closed form. So you get a short distance between the 2 points. As you increase the force a little bit, then you get a brief excursions to larger expansion, meaning it unfolds briefly and then it folds back fairly quickly. And if you increase the force even more, then it spends a longer time and in the unfolded state and or more frequently. And until now, it spends almost all its time in the unfolded state. Okay? So you can measure how the rate of transition between the closed and the open states. So if you think about two stable states with an activated state in the middle, then you can actually apply force to tilt the energy landscape in a certain direction. And then by measuring how the rates change with force, you can actually, in this case, obtain information about the distance from one state to the activated state. And in fact, if the distance is very short, then when you apply this external potential, the energy difference is not going to be very large because of small distance. So the force will have a very small effect, right? While if you have a large distance here, then the force will have a much larger effect. So based on the force-dependent measurement of this rate, you can obtain information about the energy landscape. And in fact, Block Lab has mastered this technology to extreme to really get really fancy information about not only about the position of the barrier, but also the shape of the energy landscape and so on. And in those measurements, what is essential was to eliminate the drift of the instrument that you see here. So in this measurement done many years ago, that the length of
Is kinetics full exponential? Is this a Poisson kind of data when they analyze, just treat it as a single variable?
That's right, yeah. They show us a histograms, it looks exponential to me, but they don't have hundreds of stuff, they don't have too many data points. So the drift that you see here is actually measurement effect. Your temperature is fluctuating, your instrument is drifting, and that gives you noise. And that actually really is a problem if you want to obtain this data for a long time, to obtain lots of data. Right? So the new development that I illustrate next by the Block Lab, and also later by Yang and Bustamante, actually made it possible to do this measurement for a long time scale with minimal drift. Then they could get much more detailed information about the energy landscape. So this is the idea of improved resolution instrument. So if you have one optical trap and the other is stuck to like glass surface and so on, then your laser beam may drift relative to the microscope because they are mechanically, they are not mechanically like identical. So then any kind of drift between the laser beam and the microscope will lead to noise. And they avoid this by going to two traps. You have two laser beams trapped with the two particles at the same time, but they are coming to the same laser. So if the laser beam drifts relative to the microscope, you don't get any noise due to that. So they could actually get much better resolution. So I think this movie is an illustration of the transcription reaction by a single RNA polymerase. So RNA polymerase is attached to one bead and is pulling in the DNA during transcription, making the DNA shorter and shorter. So the two beads come close to each other as a function of time. And you can determine the center of the beads again much better than the side of the bead with a nanometer precision. In fact, in this case, about an angstrom precision. So they were able to actually see the RNA polymerase in action with a single base pair resolution. We knew that the RNA polymerase would actually incorporate one nucleotide at a time. So we must move in one base pair step, but to see it actually do it is pretty amazing. So using this instrument, they were able to show that the distance between two bases is changing in 3.4 angstrom steps. Yes?
So when they do that experiment, here is, I don't understand these details, but when they do that experiment, only one bead moved. Why? But why does the RNA polymerase sit attached here and not march pull the DNA? Marching along.
So I want to explain to you, I don't do this measurement, so I don't know the full details, but I mean, in general, what you do is that you have two tracks that are different in stiffness. Okay, so one track is a lot stiffer than the other. Okay, so the movement occurs mainly in one bead. That's one way of doing the measurement. I think that's what they did.
So how do you decrease the stiffness if you're using
laser.
If you're using the same
Where split, or you can split the laser power, unequally. Now the Shemna-Bustamante technique that I published in PNAS like 2 years ago is different, where they don't need to use two different laser powers, two different stiffness. What they do, they measure both beads movement at the same time. Okay? And then their idea is that you can break down the movement into two components. One is that the movements that happen in the same direction versus in opposite direction. Okay? And then they can focus on the movements that occur relative to each other, because this one could be due to the laser drift. Right? So they could actually recover more accurate information about the distance between the two using that method. And so in this case, you have the resolution, you can see the steps, but what did you learn, that you didn't know before? Nothing. Okay? But once you have this extremely stable instrument, then you can go and do some new experiments. In this case, what they did was they studied the pausing of the RNA polymerase. It goes some distance, and then in a sequence-dependent manner, you can pause for a fixed duration and then continue. Okay? And because the instrument is very stable, they could actually measure this from many segments of the same sequence or different sequences, so that they can then realign them to see whether some of the pauses are really systematic pauses caused by the sequence versus random events that are not dependent on the sequence. Just the fact that they can really align them with single base pair resolution was really very useful in those non-trivial investigations. And please ask me questions if you have any in the middle, because I don't have to finish my talk. Okay?
What you have been doing is you have until 10 o'clock.
Yeah.
And what we have about 20, 50 minutes in place if you like.
Okay.
You can stop and let people go over questions. But people should be free to stop in the middle. But
yes.
So, and we've done a lot of different varying synthesis. I'm thinking just very basically, like if you put a piano on my back, I'm going to walk a lot different than I do without the piano. So, like, if you have different loads, like, is it very different, or is it pretty similar to all the different loads that you put on these?
That's a very good point. So I think Alex will discuss this force velocity curve. So people have measured the velocity as a function of force, and depending on the mechanism of translocation of these motors, you can predict to have different shaped curves. Okay? And then from these measures, we can actually say something about this. Well, of course, what you really would like to do is, you want to make sure that when you extrapolate your force dependence data to zero force, that should match whatever is known about the enzyme's properties that you measure in ensemble without applied force. Right? And in many of these measurements, that's the case. In some of them, that's not the case because of the particular way of the measurements are being done. So for example, in transcription, yes, this works very well. If you extrapolate the zero force, then you get the same number. But if you want to study DNA hairpin closing and opening, it turns out that if you extrapolate the zero force, you're off by about 1,000-fold compared to the intrinsic dynamics. The reason is that hairpin opening closing is a very fast event normally, and but your bead, a large bead, and the DNA handle do not equilibrate quickly enough. Okay? But that's also a very important point that I alluded to earlier. When you do this measurement, you are making modifications to the system. You are attaching your handles, fixing them to the surfaces. So in a non-measuring environment, so you always have to make sure that you don't really screw things up too much. Okay. Otherwise, you cannot publish. Okay, so next topic is the device called magnetic tweezers. So in this case, you don't actually have a laser light. What you have is a magnetic particle. It's a superparamagnetic particle attached to one end of DNA, and the other end of DNA is attached to a surface. Then on top of the sample, you bring in a permanent magnet that generates a magnetic field gradient. What the paramagnetic bead does is actually it gets sucked into the region with the stronger magnetic field gradient. So it gets pulled up, and the attachment here will also restrict its movement sideways. So if you look at the bead from the top, then it will undergo a Brownian motion in XY plane, as well as Brownian motion in the Z plane. And if you bring down the magnet, then you will increase the force, then it will be trapped more strongly, and then movement will be actually more suppressed. And by how much the movement is suppressed, you can also deduce the amount of force that you are applying. So this is an example by Case Decker's lab, where they use DNA translocating enzyme that is somehow translocating on the double-chain DNA, forming a DNA loop. And when it does that, then the DNA end-to-end distance becomes shorter and shorter as a function of time. So you can see DNA becoming shorter and shorter gradually, and enzyme suddenly releases DNA, going back to the original length, and then does it over and over again. So you can look at the data zoomed in, and the length is changing due to translocation. You can measure the speed of translocation, and you can also measure, on average, how far the enzyme moves before falling off from these measurements. This measurement was done on a DNA that has no torsional constraint. Double-chain DNA has torsion. You cannot fully rotate it around the central axis because of the torsion. But two ends of the DNA were attached to the beads and surface so that they can swivel around freely. So you don't have to worry about twisting of the DNA in this measurement. But in other measurements, you can actually utilize the twisting effect. Let me skip this one. But essentially, by measuring the Brownian motion and using the equiprecision theorem, you can gain information about the force being applied to the bead. So this is a nice example. If you measure the center of the bead and then plot the density plot in this case Z and Y, then at a very low applied force, this is like 40 femtonewtons of force, then you see a large cloud because of large Brownian motion. But as you lower the magnet and increase the force, then the cloud becomes smaller and smaller. Okay? And from these measurements, you can use this variance of the position and to get the force value of your DNA. So this is a fun part. Maybe it will work. Let's see. It works better with an Ethernet cable. Okay. Any volunteer who wants to help?
Come on, the volunteer. Come on, someone steps up.
Victor. No. It's okay.
Come on.
I'm not going to eat you. Yes. Okay. So this is a double-strand DNA, and we're going to twist it. Okay, so I'm going to twist it, you hold still, okay? So I'm going to twist it. No, I said you hold still. Twist it, and then at some point, oh, okay. So you build enough tension that polymer decides to loop out, okay? And that's called platinum, shown in this cartoon here. If you continue to twist, then you can get more of them being looped out at the same location. Okay? So you're converting twist into the so-called supercoiling of DNA. And this depends actually on the force applied. Okay? I'm not changing the twisting anymore, but now let's apply a stronger force. Okay? And then if you release the force, it goes back. Okay? So if you twist, under a fixed force, if I increase the twist, then the DNA length will go down. Okay? But if you increase the force, it'll go back up.
We all know these annoyances of our garden hose.
Oh, yeah. That's right. Thanks a lot. Yeah.